Hello. In this lecture, we're going to be covering the visual system, which allows us to process all visual information in our environment, from lines to colors to shapes to complex patterns. The visual system starts with the eye. For the information to reach the brain and be processed, the neurons in the eye must convert light energy, or photons, into neural signals. Thereafter, through pathways in the brain, it will be possible to integrate information from different neurons in order to produce a meaningful image that contains all of the separate information, like information about color, shape, or angles. So we'll start at the beginning, at the eye. So the eye is a sphere that contains fluid, and it consists of 3 layers. It is also divided into an anterior part and a posterior part. The cornea and the sclera form the outer cover of the eye, and the cornea is transparent in order to let the light pass through. The pupil is the opening into the eye, and the iris is responsible for either dilating or constricting in order to alter the amount of light that can enter through the pupil. The lens is a very important structure because through the lens, light will be refracted to the back of the eye. The middle layer of the eye consists of multiple layers, including muscles, and the choroid, which is the layer that contains blood vessels that can nourish the eye. Finally, the neural part of the eye is found at the very back of the eye, and it is called the retina. The retina contains an area called the macula, and this is the spot with the highest visual acuity, and the fovea is at the center of the macula, and it's the spot with the maximum visual acuity in the whole retina. All the axons from the neurons found in the retina exit the eye in a nerve bundle, which is the optic nerve. The optic disc is the only blind spot in the eye because it is where the axons leave the eye, so therefore there is no innervation at this exact point. So this is just a summary of all the definitions that I just went over in the previous slide on the diagram, so this is for you to make notes of. The retina is going to be our focus since it's the part that's actually responsible for producing the neural signals that are gonna then go to the brain. So there are 5 types of cells in the retina. The photoreceptors, bipolar cells, and ganglion cells are the main cells, and the horizontal cells and the amacrine cells have more of regulatory functions, and they participate in local circuits that alter the activity of the 3 major cell types. The retina is also organized into 5 layers with 3 layers of neurons and 2 plexiform layers that contain the horizontal cells or the amacrine cells and the connections between them. In this diagram, the front of the eye is on the bottom and the back of the eye is on the top. So at the top of the diagram, or at the back wall of the eye, there is the pigment epithelium that sits on top of the choroid. The pigment epithelium plays an important role in supporting the function of the photoreceptors. It removes old discs from the outer segment of photoreceptor cells, since these discs only have a 12-day lifespan, and then they can also regenerate photopigments for the photoreceptor cells after they have been exposed to light. In addition, the capillaries that are present in the choroid are needed to provide the photoreceptors with nourishment. So I told you that there are 3 main cell types in the retina, and this is because these 3 neuron types form the main pathway that is needed to send visual input messages to the brain. The light hits the retina and goes all the way to the back where the photoreceptors are. The photoreceptors then pass the message onto the bipolar cells and then to the ganglion cells, and then the axon of these ganglion cells leave the eye through the optic nerve. So this is the order of cells. Now the photoreceptors that I mentioned before, so this is where you can see the discs that we were talking about. So these cells are special because they contain pigments that can absorb light, and this is how they can convert information about light energy into neural signals. So there are 2 types of photoreceptors in the eye, rods and cones. They look similar in that they both have this outer segment that contains the discs, and this is where the pigment is found. The inner segment consists of the cell body, the axon, and the synaptic terminal. So you can already notice that they look different from the typical neurons that we've seen before. So light actually consists of waves, and different colors have different wavelengths. So visible wavelengths are between 400 and 700 nanometers. So rods respond to light across this entire range of wavelengths, while cones are actually color specific, and accordingly each cone can respond to either blue, green, or red wavelengths. That's why we have multiple. So these are the differences between rods and cones. Rods are more sensitive in the sense that they can respond to just one photon of light, whereas cones require more photons in order to be activated. Rods are a lot more abundant in the retina than cones, since almost all cones are concentrated in the fovea. Rods prefer dim light, so they work best in this type of light, while cones, since they respond to color, prefer bright light. Since rods respond to all visible wavelengths, there's only one type of rod, but cones respond to one of three colors, so there are three types of cones, each for the different wavelength that corresponds to blue, red, or green. So what is the role of the rods and the cones? Actually, without them, there would be no response to light because they have the ability to carry out a special function called phototransduction. So this means that they can convert light or photons into a neural signal. So as I mentioned, they have the pigments in the discs in their outer segment. So when these pigments absorb light, they undergo a change in their structural conformation. So this change leads to the activation of an intracellular signaling cascade that will result in the closing of sodium channels and the hyperpolarization of the rod or cone. So this might sound weird because in other systems we usually see that activation or a stimulus leads to depolarization or the production of an action potential, but here it is the opposite. So this feature is unique to the retina. Basically, when it's dark, the photoreceptors are depolarized, so this is their default setting when there is no light, and they have a potential of around minus 40 millivolts. In the light, there is an efflux of potassium ions, and coupled to the closing of sodium channels, this leads to hyperpolarization. We don't really know why the pattern is reversed in this system compared to others, but it might make a little more sense in the next slide. So the connectivity patterns in the retina are rather complicated because there are many different parallel circuits and interconnected cell types. The photoreceptors synapse onto bipolar cells. This is where the hyperpolarization of the photoreceptor comes in again. So some bipolar cells are called on-center and they are activated by light at their center, and others are off-center, which means that they are activated when there is no light in the center. Each photoreceptor is connected to an on-center and an off-center bipolar cell. So in the dark, when the photoreceptor is depolarized, it releases the neurotransmitter glutamate at the synapse, and this hyperpolarizes the on-center bipolar cell and depolarizes the off-center bipolar cell. When a light falls on the center of a photoreceptor, the photoreceptor is hyperpolarized, so it releases a lot less glutamate, which then allows the on-center bipolar cell to be depolarized and the off-center bipolar cell to be hyperpolarized. So basically, the retinal ganglion cells are the only cells that will actually produce action potentials instead of graded potentials or just small changes in membrane potential. So they are the output of the retina, and their axons are the ones that leave the retina to go to the visual processing system of the brain. The receptive field of the cells represents the area on the retina that, when it is stimulated, will affect the ganglion cell's membrane potential. There are also two other cell types that are interneurons, so they are more involved in horizontal circuits and fine-tuning, as I mentioned, and they are the horizontal cells and the amacrine cells. So we saw that the horizontal cells in the side of the synapse between the photoreceptors and bipolar cells are between those, and their function is to interact with those two cell types to fine-tune sensitivity to different light intensities. The amacrine cells are found between the rods and the retinal ganglion cells, and they contribute to direction selectivity. So once the signal leaves the retina, its first stop is the thalamus, like with the other sensory systems that we went over. The information from the retina will enter the thalamus and synapse in a specific region called the lateral geniculate nucleus, or the LGN. You might notice from the diagram that there is a point behind the eyes where the axons cross. So this is called the optic chiasm, and I'll come back to it in a few slides. After the LGN, the information is transmitted to the primary visual cortex and the occipital lobe. Now, it's important to note that this is the main pathway for the visual information, but there are also alternate pathways. For example, retinal ganglion cells can also project to other areas like the midbrain for specific functions like reflexes and the control of head or eye movements. This is just a reminder of where the occipital lobe is. So V1 is found in the back of the brain, and V1 is also referred to as the striate cortex. We saw that some axons cross at the optic chiasm. So actually, axons from the inner part of the retina, which we call nasal because it is close to the nose, actually cross in the brain at the optic chiasm and synapse on the LGN of the opposite side. This means that this part of the visual field in the left eye is processed by the right side of the brain and vice versa. Now, the axons from the outer part of the retina, or the temporal part that is closer to the ears, are processed ipsilaterally, so they do not cross in the chiasm. Now, in the LGN, there are also multiple layers that are organized in a left and right manner, so each layer receives information from one eye. There is also spatial conservation in the patterning, which means that different points on the retina correspond to different points within the LGN, and this is referred to as retinotopy. This is found in the LGN and also in V1. Now, from the LGN, the next neuron will synapse on the primary visual cortex, and this is the first point where information from both eyes is integrated, and it becomes possible to form a complete image. The visual cortex is perhaps one of the most special systems in the central nervous system because the neurons in V1 have very particular sensitivities. So many experiments were done, and a lot of those were by complete accident. So if you search 𝐬𝐬 and 𝐻𝐽𝑠 experiment on YouTube, you can find the video of a really interesting and serendipitous experiment that was performed on recordings from neurons in V1 of cats while they were observing specific visual stimuli. So this and other experiments led to the conclusion that neurons in V1 can be selective to orientation, so lines at different angles, direction, and color. Neurons with the same stimulus selectivity are organized in columns. So for example, all the neurons that prefer horizontal lines are in one column. We saw before that the cortex is usually organized in 6 layers, and here this is also important because the input to and the output from the visual cortex is also in different layers. For more complex processing and producing meaningful information from visual stimuli, there are second visual cortices like V2, V3, V4, and V5. There are also visual association areas which can process and contribute additional information to the basic visual information, such as the parietal and the temporal lobes. For example, these may include more complete representations of moving objects, color sensitivity, information about the depth of an object, recognition of certain elements in the visual field, and the integration of memory with objects that we can see. Now, for these higher order processing functions, we divide the pathways into 2 separate streams. The one that goes to the parietal lobe is called the dorsal stream, and it can answer where questions by integrating spatial information. And the ventral stream goes to the temporal lobe and is more responsible for visual recognition, and it will answer what questions. Actually, here I wanted to mention some of the most peculiar neurological diseases related to the dorsal and ventral streams because they really show why we need these association areas in order to produce meaning behind visual information that goes beyond the basic features of just vision, such as color and shape. The first one is called left neglect, and it is due to damage to the right parietal lobe, which is responsible for spatial information and attention. So people with left neglect cannot pay attention to the left side of the world. Now, it is weird because there is no damage to their vision, so they can technically see with their eyes, but their brain does not really see the left side. So this means that they will often only eat the right side of their plate. They will have trouble turning their head to the left side because they're not sure what the left side is. They can draw only right side of images, like a clock, for example. They'll only include the right side of that. And for this also, you can check this on YouTube, and there are really interesting videos and images of patients who have this disorder. Now, the last example I wanted to give, this is of temporal lobe damage, and it is called agnosia, and this is the inability to recognize or name objects, and it is due to damage to the temporal lobe since it's responsible for object recognition. Now, people with agnosia are able to process the details of an object, like its color and shape, so they're completely able to describe it or even draw it, but they're unable to recognize it and link it to a certain name or category. Now, a specific type of agnosia is prosopagnosia, and it is the inability to recognize faces, specifically. Now, there's a book called The Man Who Mistook His Wife for a Hat by Oliver Sacks, and I highly recommend it if you're interested in such diseases. Hi again. The next system we'll be covering is the auditory system that makes it possible for us to hear sounds, be able to identify their characteristics, and also pinpoint where the sounds are coming from. Let's dive in. The auditory system processes sound in the form of sound waves that are received by the ear. Sound waves have various characteristics that the auditory system can recognize, such as the amplitude, which is correlated with how loud the sound is. We can also perceive the frequency of a sound, which is its pitch. And the range of frequencies that we can actually hear is limited. For example, humans can hear sounds with a frequency up to 20 kHz, while dogs can hear frequencies up to 44 kHz. So this is species dependent. Sound waves also have a phase, and this is used to locate where sounds are coming from. The auditory system starts at the ear. We can divide the ear into the external ear, which is the part that we can see on the outside, the middle ear, and the inner ear. In the middle ear, there is the eardrum, which we commonly know, or the scientific name for it, which is the tympanic membrane. And it is attached to small bones called ossicles, of which there are 3. These are the malleus, the incus, and the stapes. The inner ear actually houses the major structure of the auditory system, which is called the cochlea. The nerve containing the sensory receptor axons for the auditory system is called the cochlear nerve. Now that you know the structures and the anatomy, I can tell you their functions. The external ear doesn't play a huge role, but its main function is to amplify the sound waves that arrive to it by up to 100-fold. The middle ear is important because this is where the eardrum is located, so the sound waves will really focus onto the eardrum, and the eardrum can collect these vibrations and pass them onto the ossicles and then to the inner ear. Amplification occurs here too. Now, one additional feature in the middle ear is that there are 2 muscles that have the important function of protecting the ear from loud sounds. This is called the attenuation reflex, and basically the muscles that are found here can contract to make the ossicles stiffer so that they will not vibrate as much, and therefore a lot of the loud sound will be blocked instead of transmitted to the inner ear. The cochlea in the inner ear contains the sensory receptors that will be activated in response to sound, and accordingly it is responsible for converting or transducing the waves into a neural signal that can then be transmitted through the cochlear nerve into the central nervous system. The cochlea is a specialized structure. It is made of 3 fluid-filled canals that are coiled into this shell-like structure. Actually, the canals are also important for the vestibular system, which is meant to maintain our balance, especially during movement. But we will be focusing on the part that looks like the snail shell, and that is the part that's responsible for the auditory system. If we cut through the cochlea to see its internal organization, we can see the 3 parallel canals. The top one is called the scala vestibuli. The bottom one is called the scala tympani, and the middle one is called the scala media. Now, the type of fluid that is found in these 3 canals is not the same one because the scala vestibuli and the tympani contain a fluid called perilymph, while the scala media contains endolymph. We will not be covering the differences between these, but I wanted to mention it just so you can know that there are different fluids in the cochlea. Now, the membrane that separates the scala media from the scala tympani is called the basilar membrane. And on top of this membrane is a very important structure called the organ of Corti, and overlying it is the tectorial membrane. The organ of Corti is the place where the neural signals will actually be generated. The sensory receptor of the auditory system, the sensory receptors are called hair cells because they have these tiny projections that resemble small hairs. The projections are called stereocilia and they are embedded within the tectorial membrane. Now, this is important for how the vibrations from the sound waves are going to be converted into action potentials. Hair cells have a really unique way of depolarizing compared to the other systems that we've seen so far. So the individual stereocilia are connected to each other via something called tip links. Now these are filaments that extend from one stereocilium to the other. And these tip links are also attached to the potassium channel on each stereocilium. The longest stereocilium on each hair cell is referred to as the 𝐾𝐽𝐫𝐤. So here in this diagram, the one that is to the far right is the 𝐾𝐽𝐫𝐤. The stereocilia will move because of vibrations that travel through the canals or the sound waves, and therefore the basement membrane will move. Now the hair cell will depolarize if the stereocilia move towards the 𝐾𝐽𝐫𝐤, as is the case in the diagram, because the tip links will pull the channels open. Now, when the stereocilia move away from the 𝐾𝐽𝐫𝐤, there is no longer tension on those tip links, so the channels will not open and hyperpolarization will occur. The cochlea is capable of transmitting information about the frequency of a sound wave due to structural differences in the thickness and the stiffness of the basilar membrane throughout the cochlea. This is a diagram of the cochlea as if it were stretched out because it is easier to visualize this concept on this straight cochlea rather than the curled up cochlea. So the base of the cochlea is more responsive to high frequency sounds, while the apex is more responsive to low frequency sounds. This is referred to as place code because this phenomenon means that the cochlea basically has a spatial code for frequency, since the specific spot that is activated will tell the central nervous system about the approximate corresponding frequency of the sound. Once this information is transformed into an action potential, it will travel through the cochlear nerve to the cochlear nucleus that is found in the brainstem. The second neuron then crosses to the other side and synapses onto the superior olive. The olivary neuron then synapses in the inferior colliculus, which is a nucleus in the midbrain that is crucial to the auditory system because it integrates different auditory inputs and starts to discriminate characteristics like pitch and frequency. The next neuron synapses in the medial geniculate nucleus of the thalamus, and if you remember the lateral geniculate nucleus, that's where the information from the visual system comes in. Then the last neuron will synapse onto the primary auditory cortex. The primary auditory cortex, or A1, is located in the top part of the temporal lobe. A1, similar to most other primary cortices that we saw in the different systems, is organized in a manner that is directly linked to the spatial organization of the cochlea. So one area receives input from the apex of the cochlea, for example, and we already know that this corresponds to a specific frequency. So basically A1 is also organized by frequency of the sound. One important function of the auditory system that we really rely on heavily is being able to tell where a sound is coming from. This is very important for our survival if you think about it, since if we can't determine where a threat or a source of danger is, for example, then we will not be able to run away from it. So to localize sound, the auditory system relies on the differences between our two ears. So as you can imagine, if a sound is behind you and to the right, then your right ear will perceive the sound first, and the brain will understand that this difference between the signal that was received from the right versus the left ear means that the sound is somewhere towards the right. So this information is analyzed by neurons in the superior olive. Two possible methods are possible, and it depends on the frequency of the sound. So the example I just gave is what is done with a low frequency sound. So the time delay between one ear signal and the other is used to determine where the sound is. But for high frequency sounds, the brain instead uses differences in the intensity of the sounds, the sound that is perceived by each ear, in order to estimate the location of the sound source. Finally, I wanted to mention a special part of perceiving sounds, which is language. So as humans, many of the sounds we hear are actually words that form speech. Accordingly, there is an area in the brain called Wernicke's area, and it is specifically dedicated to the perception and comprehension of speech. It is also located in the temporal lobe right behind A1, and it has extensive connections with A1 since it requires the input of the basic sounds to integrate that with speech knowledge in order to allow us to understand what is being said to us. It is found on the left side of the brain, actually, so damage to the left temporal cortex may lead to what is called Wernicke's aphasia, and this is an inability to comprehend speech in particular. Because of this deficit in speech comprehension, it also becomes impossible for people to produce meaningful speech.